Hey, how's it going? So I am here. It's Sunday, March 13th, 2022, and I am excited to continue talking about the Krebs cycle. Yes, good old Krebs cycle. We're in the weeds, in the deep, with the main driver of life, or metabolism in life, along with glycolysis and the electron transport chain and the transition phase between the glycolysis and the Krebs cycle, which occurs in the mitochondria. And also another thing that we should discuss is the malate aspartate shuttle. I will talk about that in a bit, but you notice with my Krebs cycle diagram here, I think last time we went all the way up to talking about isocitrate, I believe. I could have gone to alpha-ketoglutarate, but I think I'll just stick to isocitrate. So we were kind of going through all the reactions one by one because essentially, just as a reminder, what's happening here, you start off with oxaloacetate, right? And you form several intermediates through this 8-stage process, where in the end of the day, what you get out of the Krebs cycle is the following, right? You get 2 CO2s, 1 ATP, 1 FADH2, 3 NADH multiplied by 2 because you have 2 pyruvates, right? Or 2 pyruvates which go into the Krebs acetyl-CoAs which go into the Krebs cycle. Okay, so through each of these steps, you have these byproducts generated. NADH and FADH. NADH is generated whenever there's a dehydrogenation reaction, or rather, essentially, whenever you have an enzyme which is removing a hydrogen and adding it to NAD. Okay, so through like protonic adment. And so you have also, remember, as part of these dehydroxylation or dehydration reactions, you also have CO2 being generated because we start off with acetyl-CoA, which is, remember, how big is acetyl-CoA? It's 6 carbon, right? Acetyl-CoA will let go of a carbon dioxide and will be dehydrogenated down, right, to a 5 carbon molecule, and then the 5 carbon molecule will also be dehydrogenated down and release a CO2 to a 4 carbon molecule. And then what will finally happen, there will be a substrate level oxidation which produces a dehydrogenation reaction which leads to the production of FADH2, which is also important in the electron transport chain. So FADH, you're expecting NADH, but instead you got FADH2. Good for you. We'll go into the differences there, but remember, NADH is nicotinamide adenine dinucleotide. It's important and it's used everywhere, and it's mostly used for transporting protons throughout these different cycles. Okay, so but let's go and get back to the Krebs cycle. There are so many Krebs cycle diagrams around, right? So we'll go to this diagram here. This is a Khan Academy diagram, as you can see. So you have citrate, it goes down to alpha-ketoglutarate, which goes down to succinyl-CoA, which goes down to succinate, which then goes to fumarate, then which goes to malate, and then which goes to oxaloacetate, and then the cycle continues on again. Okay, so in succinyl-CoA, right? So we had that we discussed aconitase, which is an isomerase which creates isocitrate. Okay, citrate, which is coming from isocitrate, which is a variant of citrate. Then essentially the dehydrogenase reaction that I was referring to before creates alpha-ketoglutarate. Alpha-ketogglutarate's interesting because alpha-ketoglutarate, malate, and oxaloacetate, their presence in the Krebs cycle is their presence in the Krebs cycle requires them to be brought into the mitochondria. Okay, so like and they actually alpha-ketoglutarate acts as an exchange molecule for oxaloacetate to come in. So you see how alpha-ketoglutarate is formed here? Well, alpha-ketoglutarate may be formed here, and it is eventually dehydrogenated through alpha-ketoglutarate dehydrogenase into succinyl-CoA, but some of it also leaves the mitochondria through the malate aspartate shuttle. Again, I'll discuss in a moment, in order to bring in malate. Okay, malate then actually right here, malate gets converted to oxaloacetate, and oxaloacetate actually gets converted. These two are interconverted. In the cytoplasm, oxaloacetate gets converted to malate, and then in the Krebs cycle, malate gets converted to oxaloacetate. So let's just jump over there to the malate aspartate shuttle to kind of explain more what's going on. So these are essentially being generated in the Krebs cycle, but they also are being brought into the mitochondria as well, right? Because oxaloacetate, you have acetyl-CoA, which is the active ingredient in the Krebs cycle. The acetyl-CoA has to combine with oxaloacetate. Okay, how does oxaloacetate get into this citric acid cycle? Well, it comes in from the cytoplasm, right? And there is essentially this whole malate aspartate process is a shuttle itself, right? So oxaloacetate, let's start again with oxaloacetate. It comes in, gets converted to malate through malate dehydrogenase, right? Then there's this malate alpha-ketoglutarase transporter. So remember how I talked about alpha-ketoglutarase, which is an intermediate in the Krebs cycle? That gets exchanged through this transporter with malate, and how convenient, right? And then malate in here, because malate, oxaloacetate can't get into the mitochondria on its own. It needs to be converted to a reduced form to make it through this particular shuttle a protein process here, and thus once oxaloacetate is in the mitochondria in the inner membrane, then it can go through the process, right? But then also, let's say how does oxaloacetate get outside of the transporter? And you might even be asking, like, why? Outside of the mitochondria. You might be asking, why is oxaloacetate even needing to leave the mitochondria? Well, that's a whole good question, right? The different discussion, but in order to leave, it can't breach this barrier, so it has to be converted to something that can breach this barrier. Why wouldn't it be converted to malate? It's converted to aspartate, right, through what's called a deamination reaction of a glutamate. Okay, so this glutamate can go through is an amino acid. It can go through the membrane, right? Glutamate is deaminated, right, by aspartate aminotransferase, and the amino group is stripped from the glutamate, added to oxaloacetate, which creates aspartate, and the byproduct alpha-ketoglutarate. Alpha-ketoglutarate then can be exchanged. It can either be involved in the Krebs cycle, right, or it can be converted to what? Remember, what is alpha-ketoglutarate converted to? It's converted to succinyl-CoA, right? So succinyl-CoA of alpha-ketoglutarate is the 5-carbon molecule. Succinyl-CoA, remember, is the downstream element, and let's just go back to what succinyl-CoA is. This is the formula for succinyl-CoA here, right? So it's got it's essentially got a thioester bond. This sulfur acts as a thioester bond between coenzyme A and the succinate part, right? Succinyl acid part, which is 1, 2, 3, 4 carbons, right? This is succinate. So then that's the 4-carbon molecule, and then you have fumarate, which is essentially a fumaric acid. It's an intermediate between malate and succinate. So because eventually this CoA gets stripped off. But anyway, not to divert too much. So we were talking about alpha-ketoglutarate leaving, and so then aspartate is formed. Aspartate then gets finally exchanged with glutamate. That's essentially glutamate can kind of get into the cell, right? And glutamate can get deaminated outside of the cell in the mitochondria in the same process, right? You have alpha-ketoglutarate, you have aspartate aminotransferase, and then you have that essentially reducing the aspartate into oxaloacetate. So transferring the amino group from the aspartate to the oxaloacetate again outside. So basically that's what's happening there. Now we eventually want to talk about what fumarate is. Fumarate's interesting because the presence of fumarate in the Krebs cycle, fumarase here, right? Fumarate is converted to malate through an enzyme called fumarase. Okay, so we'll talk about this, but fumarate actually is indicated. It doesn't seem that important a reaction, and it's ultimately not so important other than its byproducts, which we were talking about before, right? Like we know that ATP gets generated at some point here, right? When succinyl-CoA gets converted into succinate, right? We have this GDP turning over to ADP. This GDP actually is involved in the production of ATP through something called substrate oxidation, substrate level substrate level oxidative phosphorylation, right? So you have this substrate molecule. GDP is the substrate. It's a molecule that contains so essentially we strip, right, phosphoryl group from the succinyl-CoA. We add it to GTP. GTP then adds that molecule, right? Because GDP interacts with succinyl-CoA synthesase to result in GTP. GTP then interacts with another phosphorylation, another kinase, which then strips GTP, which is the substrate, and adds that phosphoryl group to ATP. So that's substrate level, GTP being the substrate level oxidative phosphorylation. Okay, great. So many words, I'm gonna get it right one day. So that's essentially what happens, and then fumarate, I was talking about what it's indicated in. It usually is indicated, there's certain brain tumors, neuroblastomas, usually benign tumors, I believe, are a result of a lack of the ability to convert fumarate to malate. So we have to be careful of that. There's also other places that when if aconitase, for example, is inhibited, then you can have worse issues. You can have actually brain gliomas, which are cancerous brain tumors. So curious as to why the brain is affected by this cycle so much, but that might be only one of the medical complications of this cycle. I will go tomorrow into malate and oxaloacetate a little more. It might not be worth going too much into the specifics of these enzymes because they kind of behave similarly to the enzymes we've seen so far that perform dehydrogenation and isomerization, right? So it might be worthwhile to go into succinate dehydrogenase because that has a unique compound. So anyway, that's it. I will talk to you later. Like, subscribe, and comment below. I have to answer the phone, and I'll see you next time.